Okay, thanks folks for getting up and getting here early. And today we have Dr. Grossman, who is a professor of oncology from the Kimmel Cancer Center and is the co-leader of the Brain Cancer Program and has directed the clinical and research neuro-oncology efforts at Johns Hopkins since 1981. And Dr. Grossman's expertise is in brain cancer, translational and clinical research, and directing large multi-center, multidisciplinary clinical trials and training programs. He received his undergraduate education at Harvard, his MD at the University of Rochester, his internal medicine house staff training at Strong Memorial Hospital in Rochester, New York, and his oncology fellowship at the Johns Hopkins University School of Medicine. And he is the past president of the Society of Neuro-Oncology. So thank you very much for coming today.
So it's a privilege to talk to you, and I prefer to keep this sort of informal, so if we're going along and there are questions, please feel free to let me know. So what I'd like to do is to talk a little bit about sort of why we're really interested in early phase clinical trials and how you can get into trouble doing clinical trials. And it turns out that both are pretty easy. It's easy to get really interested and it's easy to get really in trouble. And rather than sort of make this a very didactic lecture, I thought what we do is actually use examples of how we can get into trouble and maybe what we should do differently. So I'm assuming that you guys are not experts in glioblastoma, and I'm going to use examples from brain tumors. So we're going to spend just a few minutes talking about glioblastomas, sort of what are the standard ways in which we develop new drugs, and then we'll move to examples. So it all starts and ends with patients. This is an example of a 53-year-old who was really perfectly well until developed a seizure, came into the hospital. In retrospect, it had a little weakness. Has these lesions in the brain where you can see this crosses the corpus callosum to the other side. In another area here, these light up on a contrast-enhanced MRI scan because the blood-brain barrier is leaky here, so the dye leaks out. It's not a really good marker of the edges of the tumor, but it's what we use to be able to see that there's something wrong there. The patient gets started on some steroids to reduce the swelling and gets put on seizure medicine, so he doesn't have another seizure. He undergoes surgery and comes up with the diagnosis of glioblastoma. And in about 3 or 4 days, he's gone from being totally healthy to being somebody who's got a terminal illness. It's a big jump for patients. And then they come to see us and ask what should happen there. So we don't know why these patients develop glioblastoma. It's not a disease that typically runs in families. It just happens out of the blue. There are problems with this. The tumor has no capsule around it, so you can never completely remove it with surgery. So the cure rate of surgery is 0. You're limited in terms of the amount of radiation you could give because you would have toxicity to the normal brain. The chemotherapy has trouble getting in because there's a blood-brain barrier that is meant to protect the integrity of the nervous system and allow your synapses to continue to fire the way they're supposed to. Even if you eat, drink, smoke, do whatever else you're going to do, the brain is pretty stable. And the imaging that we use is really not like imaging where you have a chest x-ray or a CT scan, you can see where the tumor begins and ends. This, if you take out everything you can see in surgery and your post-op scan looks great, you're going to die from the tumor. This, if you look at autopsy studies, this is a disseminated disease. It's not just in one location. With all these challenges, we haven't made a whole lot of progress. Before 2005, there was a survival curve for patients with glioblastoma. Almost everybody dead in 2 years' time. Between up until 2005, there were all kinds of studies. First looking at nitrosoureas, then looking at a combination of nitrosoureas and other drugs, and then in 2005, temozolomide became available, but none of these showed dramatic improvements. The study that actually made temozolomide the only drug that actually changes survival in patients with glioblastoma was this one, which was published in the New England Journal in 2005. Patients were randomized to radiation alone versus radiation with daily temozolomide given during the radiation, followed by cycles of adjuvant temozolomide. The plan of this study was to take survival from 12 months to 16 months as a median, so that success would be defined as adding another 4 months to your median survival. And this study did not hit those goals and still managed to get published in the New England Journal. Median survival went from 12 months to 14 and a half months. But the survival at 2 years was substantially different. And that was enough, because the bar is really low in glioblastomas. That was enough to make it. So this is standard therapy for patients with glioblastomas. The other thing that these investigators did is they looked at something called MGMT methylation. There is an enzyme that repairs damage, particularly from chemotherapy, called MGMT. And if in a normal situation, if you injure your DNA, either with BCNU or temozolomide, this MGMTS repair assay comes and fixes the problem. So in fact, it makes the chemotherapy less effective. But if your tumor is methylated, the DNA repair enzyme doesn't show up for work, so the drugs work better. And they took a look at the MGMT status in these patients. What they found is that if you were MGMT methylated, that is, the repair guy doesn't show up, you do much better. But interestingly, again, these are the methylated patients. The 2-year survival for the methylated patients is 22 months and goes to 46. The patients who are unmethylated have a much worse prognosis. Their 2-year survival is very low if they get radiation alone. It's somewhat better if they get radiation with temozolomide. And this is the 5-year follow-up on this data. You can see, in fact, only 10% of patients are alive with the chemotherapy at 5 years. The patients who are methylated do better than the patients who are unmethylated. So here's a summary of our progress. There have been a lot of studies done since 2005. Nothing has worked. So the standard of care remains radiation plus temozolomide. Median survival goes from 12 to 14 and a half months. 2-year survival was increased, but everybody still has a tumor. So if you're in my shoes, giving people radiation and temozolomide, although it's the right thing to do, it's not anywhere close to good enough. So what we have to do is actually have to come up with better treatments. And the question is, how do we do that? Surgery is not going to improve. I mean, people have worked hard on trying to improve surgery, but this is not a surgical lesion. Radiation, you're limited by the amount of radiation you can give to the adjacent brain. And as I mentioned, this tumor has little fingers that extend deep into the brain, so the volume of radiation you have, the volume of brain you're going to have to radiate is pretty large. So drugs are obviously what we're really interested in. Typically, we think of drug development in 3 phases. Phase 1, does this drug, what kind of side effects does this drug cause? What are the doses that you can get up to before you develop problems? What's the maximum tolerated dose that you should take on to the next study where you're actually going to look at efficacy? You'd like to know something about the pharmacokinetics, pharmacodynamics, which you get from this. And usually what you do is you take small groups of patients, you give them dose 1, they all do well. A month later, the next group gets dose 2, the next group gets dose 3, until you get into trouble. So in fact, if you think about it, the goal of your study, phase 1 study, is actually get people in trouble. You're going to dose the drug until you have a problem, then you go down 1 dose and call that the maximum tolerated dose. The phase 2 studies, I think, are the most fun and the most informative. You want to set up a trial to figure out whether this drug that you now know how to give, and you know what the right dose is, but you want to find out if it doesn't work. If it doesn't work, we should drop it, do something else. If it looks like it might work, then you want to take it on into larger studies, which are usually the randomized studies. So we'll talk a little bit about how we would do this. So many of the drugs are drugs that are out there. They work for lung cancer, they work for colon cancer, and you say, well, would this work for brain tumors as well? You probably don't need to do phase 1 studies for that. You already know the dose, you know the PK, you know how the drug is given, so you really can think about moving this directly into efficacy trials. There are a bunch of other projects where you might start in the lab and say, look, I think brain tumors have a vulnerable place right here, or a particular mutation right here, and that we would do best to develop a sort of brain-specific new drug, in which case you have to look at the pathways of basic science behind it, you have to look at screening in animal models, look at animal toxicology, and then you need to bring it in and do that phase 1 study before you can actually jump into phase 2. So it doesn't surprise you guys to say, well, that's a lot of work. I'm just going to stick with these drugs here and let, I'll just go straight into the clinical trial. So this may be the important slide of the day, if you will. If you're going to do phase 2 trials, what you want to do is make sure that you do it right. So each agent that you study, you need to set up the trial so that you maximize the chances that this drug is going to look good. You don't take patients who are desperate because they're not going to do it. You pick patients who you think are going to show off your drug the best. If you can't make it work in that situation, the rest doesn't matter. So oftentimes people get into this discussion, well, I want it to be generalizable to everybody. Down the road, you will want it to be generalizable to everybody. But when you start, you want to make sure that, in fact, you're setting this drug up so that it will succeed, if it has any rationale. So you need to make sure that you've got all your preclinical data. You don't want to spend a lot of time working on a drug where you never show drug forwards anyway. So this becomes a critical part. In addition, you need to make sure you get the drug to the target. If you don't get the drug to the target, if it doesn't reach the tumor, for example, then you are wasting your time. And as those of you who've been involved in clinical trials, I'm sure will agree, this is kind of like having children. These trials, you start them and they don't go away. You're with them for years. You're dealing with all the problems that come up with them. So if you're going to do a project, you want to make sure, number one, it's a project that you're really interested in. Number two, it's doable. Number three, it's something that you can see having some potential down the road. Or there's a question that needs to be answered. I'm okay with doing a trial and saying this doesn't work. We answer the question. Declarative statement at the end of the trial. So you have to make sure that you're getting the drug into the body the way you'd like to do it. You got to make sure you get the drug into the tumor. Sometimes you decide that you're going to put the drug directly into the tumor. Then you know it got there. Well, maybe. We'll talk about that. And then you need to make sure that you do your dose escalation. You don't want to try this at a subtherapeutic dose. It's not going to work. You could have the right drug, but you could have the wrong dose. In addition, afterwards, when you move to the efficacy trials, you need to make sure that you set up your patient population properly, and you're looking at outcomes. You need to make sure that the endpoints of your study are things that you can live with down the road. They can't be questionable endpoints. You're going to get up behind the podium, present the results, and do anything else. So we're going to walk through this little thing one at a time. We'll pretend that we've done all of this stuff, and let's take a look at delivery issues, and we'll do this by some case examples. So this was a study that we did, and as you'll notice, I'm picking on myself. These are studies that we've done. In retrospect, we might have done things differently. So we used a drug called Taxol, which is in breast cancer, colon cancer, lymphomas, and we picked the dose of the drug that has been well established in other tumors. We didn't have to do the early phase work. And it turns out that in the test tube, this works really well against glioblastomas, and it's also a radiosensitizer. So great to use patients with glioblastoma. We did this before temozolomide became available. So what we decided to do is we give these patients Taxol before radiation. These were patients who had no prior treatment except for some surgery, and they had measurable disease afterwards. And the issue is if we can't make the glioblastoma shrink in this situation where it's not resistant to all kinds of drugs it's gotten before, and it's not going to work. So we gave the drug pre-radiation, sort of a pre-radiation window to see whether the drug would work. We used the same dose and schedule that had been used in other patients. In other patients, it makes your hair fall out, the blood counts fall out, you get mucositis. In our brain tumor patients, nobody lost their hair. Nobody's blood counts fell out. We did pharmacokinetic studies. It turns out that blood levels in these patients were 1/6 of what they were in breast cancer patients. These patients were on seizure medicine, Dilantin, P450 inducer, so the blood levels were rock bottom. There is no way that this is going to work in patients with brain tumors. We never got it into the bloodstream properly because of drug-drug interaction. So it turns out that if you're not on Dilantin, the proper dose for this particular size person would be 250 milligrams. If you are on Dilantin, because we did the PK studies, it turns out that to get the same exposure, you need 822 milligrams to get the same blood level when you're on Dilantin. How big and how tall would you have to be to get 822 milligrams as the proper dose if you weren't on Dilantin? You have to be 15 feet tall and 600. Huge interaction. We don't pay any attention to that, right? So if we were going to do a clinical study with Taxol, we would have come up with a negative answer. It could be that it was a good drug, but if we give it at this dose, when we should have been giving it at that dose, obviously it's not going to work. Once you do get the drug into the bloodstream, then you have to get the drug through the blood-brain barrier. So you may have even lower concentrations that get to the brain. So it turns out that this is a major obstacle to the entry of drugs. Papers are out there showing that 98.6% of drugs on the market do not easily penetrate the blood-brain barrier. So we have to think about, before we do an efficacy trial, we have to think about how much drug do we get to the brain tumor, because if we're not reaching therapeutic concentrations, again, we're wasting our time. We should be doing another project. There are a couple ways to do this. One way to do it is to give the patient the drug before they go for routine surgery. They're going to get a surgical debulking anyway. Take the tumor out, use part of it to measure drug concentrations in the brain. The other way is the way that we've sort of pioneered, and that's using microdialysis catheters. These are FDA-approved catheters. They're usually used for people who are motorcycle accidents, sustained head trauma, looking at local concentrations of glutamate, lactate, pyruvate. This is a little microdialysis catheter. It's got a little gold tip at the end, and it can stay in the brain for weeks or longer. So the idea is that this is just a schematic that you drip artificial CSF through the tube. This is a semi-permeable membrane, a drug in extracellular fluid of the tumor. If you put it into the tumor, the drug in there would get caught up here, brought into vials. You would be able to measure the arrival of the drug in the extracellular fluid of the brain and its exit and the area under the curve. So you can do sort of intratumoral pharmacokinetics. So what we did is we took patients who were going to surgery anyway, and who gave us informed consent for this procedure. At the time that they had surgery, we asked the surgeons to put in microdialysis catheters in tumor that was remaining after the surgery. And then we gave them high-dose methotrexate in the neuro ICU 1 day post-op. We measured the plasma curves, and we measured the concentrations that were coming out of the microdialysis catheter. Here's what we found. In 4 patients, we actually found that in some patients, these curves are really parallel. This is a log scale, 36 hours out. But you can see that, in fact, the plasma curve is higher. That's what you expect than what's in the brain, but the curves are the same. But then take a look at these 2 patients. Plasma curves are the same as the others, but what's inside the brain tumor is not at all the same. So the first answer is, well, this technique doesn't work. But it turns out it does work. If you look at where the tip of the catheter is, where that little gold bead is, This is an area which is contrast enhancing. That is when you give contrast, it eats into the blood-brain barrier, so the blood-brain barrier is open. This, the neurosurgeon can't tell what's enhancing and what's not enhancing. So they put it in, and in fact, there's no blood-brain barrier breakdown here. So the drug has trouble getting in, and when it does get in, it has trouble getting back out again, showing you the importance of the blood-brain barrier. So one of the current fads in neuro-oncology is to use a drug called the Systematic or Avastin, which actually closes the blood-brain barrier. It reduces swelling and edema. Patients symptomatically improve because there's less mass effect and less edema. But what you're essentially doing is taking your delivery of drug to the brain and give it along with the chemotherapy, and you convert yourself from this curve to this curve. You basically close the door to your chemotherapy. A lot of other folks, forget the blood-brain barrier. Let's just put the drug right into the tumor. Let's put catheters in. Let's put those catheters on pumps. Let's grip the drug in. This is called convection enhanced delivery. So in this case, the catheter was placed here. Catheter was placed here. Here's the contrast enhancing mass. But just because you put the catheter there doesn't mean you're going to have good distribution. So if you put a radionuclide tracer in, and then you image, you can see that in fact you treated this area, you treated this area, but this area is totally untreated. There is no way this is going to work. If this was a radiation field, you would be subject to malpractice, because there's a large portion of the tumor that you actually never treat. So again, it just can't work. The gliadel wafers were invented here at Hopkins. They're biodegradable wafers that contain BCMA, chemotherapy drug. And the idea is at the time of surgery, you would put the wafers in the surgical cavity. Here's the survival curve in patients with gliadel wafers with a glioblastoma. There is a little difference, and it just happens to be at the 50% mark. So it looks like it improves median survival. And because the bar is so low, we have so few treatments in glioblastoma, this actually was FDA approved for patients. It's not a big difference. So what dose of the drug did they decide to put in the wafers? So they picked 3.8% by weight will be chemotherapy. Why did they pick that? Well, if you calculate the amount of BCMU that's given intravenously versus the amount that we put in the brain, this is actually a pretty high dose, and the company decided that was as high as they wanted to go. But the animal studies showed that the more BCMU you put in the wafer, the better your survival. So our brain tumor consortium decided we would do a formal dose escalation study. In each wafer that contains 3.8, there's 62 milligrams of BCMU. But we did a dose escalation study, and we found out that we could give 20% rather than 3.8% safely in patients. We went back to the company and said, okay, looks like your product had maybe a little less efficacy to start with. You could actually make this a lot better. And they said, no, we'd be competing against ourselves. It costs a lot of money to do that. It might come up and people are not going to do it. Take home message. You're going to do your phase 1 studies. Do them right the first time. Don't start your treatments with, suboptimal dosing, because once you do that, it's hard to make the difference over time. So I think we've covered sort of these issues about local delivery. Now let's talk a little bit about suitable trial design and how we would put that together. So this is your first step at trying to see whether things work. Again, we talked a little bit before about the fact that you have to be pretty careful to set that up in a way that will work. Many clinical investigators fall in this category, and if we pull this off, we're going to eat like kings forever. But it may be a little overly enthusiastic. I mean, I think the way most people get in trouble with phase 2 trials is they try to do way too much. I mean, the KISS principle really works. Figure out what your major question is, and let's answer that question and build on it sort of one piece at a time. For example, how do you get yourself in trouble? Most brain tumor trials, when you're doing your first effort. You do this with a new drug. You don't do this with people who just are getting started with treatment. You do it with people who have heard where taking some extra risks is a little more acceptable. You wouldn't say, well, I don't think you should take radiation at 10 years old. I think you should take my new drug first. My new drug has absolutely no data behind it. And when people's tumor comes back, then you can sit down and have a discussion that says, your options are pretty limited. You didn't fail the best agents we have. We have another agent. We don't know anything about it. So there's a tendency to say all patients with recurrent disease would be eligible for the trial, right? Anybody who failed, you should offer this drug to. But remember, you're trying to make a statement at the end of the day about whether this drug works or not. And depending on which patients you select, you will get one answer or another. So this is a study that we did looking at all the patients in our brain tumor consortium who had recurrent disease and what were their prognostic variables. So this is a recursive partitioning analysis, where we took a look at all the patients and then separated them out into prognostic groups. I don't know that you can read this from the back. This is initial histology, age, and steroids. So if you're old and on steroids and have initial pathology is a grade 4, you're going to do initially. Median survival here is 4 point some months. Median survival here is like 26 months. If I select just these patients, I'm going to show that the drug works really well. It doesn't have anything to do with the drug. It will all be patient selection. If I pick this group, I'm going to show that the drug doesn't work. This is not a homogeneous group of patients. Some patients are elderly, they got radiation Temodar, and they grew right through it. Those people are going to have a very short survival. Some people are younger with better molecular characteristics, and they may not have been treated for 2 years, and then you're going to put them on front, they're going to do much better. So when you're thinking about setting up your trial, and you just decide you're going to take the big puddle and put it in there, you're essentially you're asking for a result that's going to be very hard to determine at the end of the day, and that's probably not the way you want to set up your trial. Again, there are all kinds of issues with regard to individually diagnosed patients, about how you select patients, and what your endpoints are going to be. I forgot to mention that this is the survival curve from these cohorts. You could pick a group of patients down here or up there. Sure.
But regardless, you'd have a control group, wouldn't you?
No.
In a phase 2 study, no control group?
So, great question. Randomized phase 2 trials are fun, right? Half of your patients are going to get standard therapy. What's the chances that a new drug is going to work? Small. So you're going to put you can put half your patients on standard arm, and the odds are overwhelming. I mean, nothing has worked for 15 years. So if you find out that your phase 2 trial shows that there's no difference from everything that's been done before, which is the likely outcome, you didn't need the phase 2 trial. You didn't need the control. You only need the control arm if your results look good.
Yes. But you don't know what your results are going to be when you start.
So maybe doing a small study with a limited number of patients, and it's an exploratory. And if you find that there's something in there that warrants further evaluation, then you can open a randomized phase 2 that can go to a phase 3. But if the requirement is that all new drugs, every new drug that's tried, tested in a randomized phase 2 trial, 50% of your potentially eligible patients are going to get standard therapy.
So how do you know that you're getting a favorable outcome?
So survival is very short, right?
Mm-hmm.
So if you end up with patients who are surviving much longer than they should, you've got a drug that you actually need to pay attention to.
So it's versus historical or concurrent?
So we would start with historical control, good historical control. Again, I understand that I'm out on the fringes on this because the NCI and other groups are saying that everybody needs to do a randomized phase 2. I think that's nuts because you end up controlling things where there's not a hint that they work at all. And you end up, I mean, if you're running a trial and you have a randomized trial, suppose you're the patient and you say, I would like to join the study, I want to try something new. You get randomized to the control. It's like, no, I'm not going to do this. Going on to another study. It's very hard to approve a randomized phase 2. So we'll get back to some of those issues, which are good issues. Yeah. Sure.
Can you go back to the brush?
Sure.
So when you're showing that the people with the younger age and have the glioblastoma, so with the survival curve up there, is that all sometimes most likely true that you're going to select those people with higher survival for your phase 2 trial?
Yeah. So there are multiple prognostic variables. One of them is age.
So you choose the people who have the most optimal outcome maybe then.
So again, it gets back to the last question. If you're going to have an internal control arm, and you have some control of that, if you're not going to have an internal control arm, then you need to make sure that you have a historical control that is satisfactory to make that decision. If there is no historical control, then you have no choice but to add on. So let me give you an example. We'll go a little off topic for a minute. We end up with a patient who's got a particular mutation in the brain tumor. And you potentially could target that mutation with a drug. You target it, you end up with an outcome that looks better. But we don't know what the natural history of tumors with that mutation is. Could be that the natural history is that they would be better. In that case, you better have an internal control on it because you have no data about what the natural history of that tumor is. If there's another person who's done that study and you've got a historical control arm, then you're better off. So again, there's a balance here, which we've been talking about between sort of perfect and good. Sometimes you can't let perfect get in the way of good, otherwise you just don't get stuff done. But this is why this is the fun part about this topic. Yeah.
So here, instead of with recurrent disease, is it that when a drug is tested to be efficacious in a recurrent disease, then you work its way into the first line treatment?
So often you have the best chance of making a survival impact in people who've not been very heavily pre-treated. So oftentimes the early studies are done of recurrence. You figure out the PK, you see whether there's any sign that this has any activity at all. If we go to the NCI and say we want to use this drug, they'll ask us, what data do you have about activity? And if we say none, it's going to be very hard for us to justify doing this in the upfront setting. So we'll move right to your question about how to do things. Let's pretend that each of you in the audience own a drug company. You're really interested in vaccines against glioblastomas. There are no good treatments for glioblastomas. And so you say, well, I think we could add a vaccine to the radiation MR and it would really work. So you end up identifying a target specific to glioblastomas. You figure out how you can effectively immunize these patients. You study it in several tumor models. You jump through all kinds of regulatory and manufacturing and FDA kind of hurdles, and you do your phase 1 safety studies, and you're all good. So we did all of the safety studies in recurrent disease. Right now, you've convinced yourself you've got good science, it works in animals, it's safe in man, you're excited, you've published abstracts and manuscripts, you spent a lot of money, and you spent a lot of time, and now you're going to take this into clinical patients. So, you talk to your clinical investigator colleague, say, come on, help me set up a trial. And he says, we can do this, no problem. We're going to do this in newly diagnosed patients where we have the most chance to impact survival. We'll do it with the standard treatment, the radiation and MR. We'll use survival as an endpoint, nice and clean. We'll use a reference group to start with because there's a lot of randomized historical data on the use of radiation and temozolomide from modern studies. So what you're going to do is say, well, we're going to take, this is the radiation temozolomide, this is the radiation alone. We're going to look at this curve. If the answer is it's well below this curve or the same as this curve, well, then the vaccine didn't add anything. If it's above the curve, then we would go further into either a randomized phase 2 or phase 3. And that way we could do this in about 7 patients. You write the study, you get IRB approval, you accrue patients very rapidly because you're using multiple centers, and your survival endpoint is close by, and the little red dotted line is what you find. Nothing's different. So you spend a lot of time and energy, and the question is what went wrong. 2 options. Number 1 is the lousy vaccine. The stuff that was done in animals does not translate over to what's done in people. Number 2 is you got screwed by your clinical investigator. You set up a trial. Which one do you think it is? Well, we're giving you a talk here. You probably know which one it is. It's not. So what do you do wrong? So this is a study that we did in the Brain Tumor Consortium, where we looked at what happens to your lymphocytes, your immune system, with radiation and temozolomide. And these patients started out with a CD4 count that was about 600, which is within the normal range. They got radiation and temozolomide. Look at the display of the CD4 counts here. Look at the display here. Now we have 40% of patients with a CD4 count less than 200. In patients with AIDS, that's where we start prophylactic antibiotics. So this is severe immunosuppression. When these counts go down, they don't come back up. If you take a look at survival, it turns out that the people whose CD4 counts stay up live longer than the people who don't. You do multivariate analysis, and this is an independent variable. The more immunosuppressed you are, the worse your survival is. And you don't die from opportunistic infection. You die because your tumor grows. So we all have a functioning immune system, and it's working to try to work on the tumor. This is iatrogenic. This is stuff that we do. It's common, it's severe, it's long-lasting, and it's associated with survival. Should this surprise us? Well, really, no. We're all working on vaccines, checkpoint inhibitors. We believe that if you can improve the immune system, the tumor's going to be fought better. So the corollary is if you hurt the immune system, probably you're not going to do so well. It turns out that the reason for this is interesting. It's all due to radiation of circulating lymphocytes as they pass through the radiation field. They are the most sensitive cell in the body to radiation. So we are accidentally hitting the immune system. And we've recently published work showing that, in fact, checkpoint inhibitors don't work if your lymphocyte count is low. So the study was done to see if a vaccine would work. The patient population that they tested their vaccine in was like patients with AIDS. Why would you test a brand new vaccine first in like a patient with AIDS? It was unlikely to respond. So all the money and the effort that went into setting up that potential clinical trial was for naught. Yes?
So, I mean, is that population of not treated people always, do they always have a really bad immune system like that, or is that just the bad luck with the sample, or what?
So it turns out that this is true not only in brain tumor, true in head and neck cancer, lung cancer, colon, in every cancer where radiation is given. And it's just an underappreciated effect of the radiation. But the point of the story here is that if you're really going to set up a trial to look at whether your vaccine works, you need to make sure that you're doing this with an appropriate patient population. You don't pick a patient population where they are immunosuppressed because the drug is not going to work. So here's all of the preclinical work that was done to get into your vaccine trial, totally appropriate. But somebody will sit down with you before you spend all your company's money on a clinical trial and say, you know, is this really the best way to test this population? So here we are with this patient that we started with. Survival curve is miserable. We've talked about the fact that clinical trials are not so easy, but here, I'd like to now sort of talk a little bit about the promise. I mean, the only way we're going to make progress is by doing these trials. Since 1994, we've been funded by the NCI to run phase 1, phase 2, the earliest of clinical trials in patients with brain tumors. This is done with 11 different brain tumor centers around the country, stakeholders here at Hopkins. Let me just give you an example of some of the trials, and they relate a little bit to this. So the first study I want to talk about is a study looking at what we could do about these lymphocytes. I mean, if we're going to think about immune therapy in patients with cancer who get radiated, we have to think about how it is we protect the immune system from the effects of radiation. Otherwise, we're going to have a problem in 40% of the patients who we're setting up for failure to do this. So this data we've already talked about. In fact, it happens commonly. People have trouble believing this could be due to radiation. I think it's due to chemotherapy or steroids. It isn't. Here's a lung cancer study that we did in stage 3 non-small cell lung cancer. You can get radiation and chemotherapy together. And here's what happens to your lymphocyte counts. They go down and they stay down. Or you can give chemotherapy first, neoadjuvant chemotherapy, and then you can give radiation. And in that case, the lymphocyte counts stay fine until you turn on a beam and they go down. The other thing that we found is that, in fact, the primary driver of lymphocytes is IL-7. It's like erythropoietin for red cells. So if your lymphocyte count is low, your EPO level should be high to bring it back up again. And it should be the same with IL-7. These are IL-7 levels in patients who have AIDS or who got chemotherapy to drop their counts. These are the IL-7 levels in patients who got radiation. So for some reason that we have no clue, IL-7 levels are low in patients after radiation. So we just opened a protocol last week that will give patients IL-7 who have low lymphocyte counts to see whether in fact we can bring the lymphocyte counts up again. There are several other approaches to this. The IL-7 is just one. But just to give you an idea about what our endpoints are, we're doing phase 1 component. We're looking for the optimal dose of IL-7. We're trying to rule out the effects of steroids. We're looking at what happens to the lymphocyte subtypes and cytokine activity. This is an early phase study. If we show that we can raise lymphocytes, that doesn't mean that people are going to live longer. This could be a driver, it could be a passenger. So we would have to do other studies to show, that in fact, and we don't know how long the effect would last. If the counts go up for a day, it's not going to be worthwhile. If they go up for a month, you give it monthly, but this is just a way to get the thing started. This is another interesting drug we have. This is a global transcription inhibitor, and it was given to patients in India by injecting it directly into the tumor itself. This is a cancer in the mouth that had occurred after radiation. They injected it, and it totally disappeared. Looks like it works really well in tissue culture, and one of the nice things is when you combine it with chemotherapy regimens in animals, it dramatically improves survival along the board. We did a study looking at this in patients with brain tumors, giving an IV, but giving a transcription inhibitor IV. It should be there every day. Shouldn't be there for 5 days and not there for 25 days. So we developed an oral formulation, and this oral formulation is now being given to our patients. We're looking again at, can we get it into the bloodstream? Is it absorbed? What are its toxicities? What's the maximum effect? And then if this works, both in leukemias and in brain tumors, we'll be doing it as a single agent and combining with other drugs to see whether in fact we can mimic what happens in animals. We talked a little bit about that study with Taxol, about why it was so interesting to think about putting it in with radiation. But we had that drug interaction stuff, which basically resulted in us not using that kind of anticonvulsant anymore in the brain tumor patients. We switched to anticonvulsants. But also the other problem is Taxol doesn't get through the blood-brain barrier. So we have a new Taxol type drug that does get through the blood-brain barrier and is orally available. So we're going to give it just like we give Taxol, we give it every day with radiation. and we'll see what happens. So this is, we're doing the first study looking at the MTD in patients with radiation. We will be looking at sort of what happens to those patients. Again, if we get a couple patients who live much longer than other folks, we'll think that we have something, but again, this is just a dose escalation study. We're also looking at specific molecular markers on the tumor. Turns out that about 3% of our gliostomas have a BRAF V600 mutation. Again, we've got a couple papers that have been published showing that in fact there are case reports that this might work, but we don't know how much drug gets in, so we're going to be studying that, and we're also going to look at this in a formal way to look at both response rates and duration and resistance pathways. One of the problems that we have in the brain tumor field is that because the drugs don't get in, we tend to screen tumors for mutations, but you can't get the drugs that will work in those patients to the target. So we have to deal with the issues of the blood-brain barrier. And this is a new study that we are proposing to the NCI a week from tomorrow, to the Brain Malignancy Steering Committee, which is really cool. It may or may not work, but somebody has to do the study, so we're going to do it. There's a lady up in Cornell who's been looking at adenosine A2 receptors, and she found that it actually opens the blood-brain barrier, that these drugs close the P-glycoprotein pump when they allow lymphocytes to get in. So it looks like a local repair mechanism for the brain, where it can sort of, if there's something wrong, we can have the local fix. In rodents, if you give regadenoson, you can see that the concentration of a large dextran, 70 kilodalton dextran, it normally doesn't enter the brain, goes in, both in rats and in mice. Interestingly enough, the concentration that's required in mice is 0.5 milligrams per kilogram, and the concentration in rats is 0.0005 milligrams per kilogram. There is a dose, it's a receptor-mediated thing, so there is a sweet spot. If you give too little or too much, it doesn't work as well. We've now looked in rats and shown that, in fact, it doesn't change the plasma pharmacokinetics, but it does change brain entry and the brain-plasma ratio. So the question is whether human blood-brain barrier is really similar to rat blood-brain barriers. This drug is a drug that we use in patients every day at hospitals as a chemical stress test for people who can't walk on a treadmill. So we are now looking at this in patients. We have two studies that we've done in patients. One where we've looked at, can you get more contrast dye for MRI scans into the brain? And one where we've used the microdialysis catheters. And so far we have not been able to show that we can increase concentrations in the brain. But the only dose we've used is a dose that is FDA approved for cardiac studies. So we are now proposing that we test all of the safe doses of regadenoson, and that we will give these patients regadenoson and then contrast. And we'll look to see whether in fact we can open the blood-brain barrier in the areas that are way on the other side of the brain, so this would be a normal blood-brain barrier, or whether we could open the barrier around the tumor or inside the tumor to get more drug in. Again, one of those things that if we can show we can increase the barrier, think about it, we could take patients with breast cancer or lung cancer or at high risk for brain metastases. We could send those patients to our cardiologist and say, please do a regadenoson cardiac stress test. They would come back to you and say, it's fine. You'd say, great. We're not going to take that drug to our oncology infusion area. We're going to give the drug along with the chemotherapy so that the drug will get into the brain the same way or similar to if it gets into the lung or the liver, and maybe we can actually prevent people from getting brain mets. Or maybe we can figure out how to get some of these drugs that might work against our cancers into the brain where now they don't get into the brain. So I'll stop we can have some time for questions, but this is sort of the way it is. The people in the lab, like the guys who developed your vaccine, are sure that they did all the heavy lifting. And that, I mean, I got you the right product. Giving this to you as the clinical investigator, it should be a piece of cake for you to make this work in patients. I lift, you grab, is that concept a little too hard? What he's saying. It doesn't work that way. I mean, there are challenges on this side about making effective compounds, but there are big challenges on this side from the clinical investigation thing about setting up your trial right, making sure you conduct it right. Otherwise, it doesn't work at all. So the end of the story is it takes a team. Everybody's got to pull it together. So with that, I'll stop. Glad to answer any questions you have. Yes?
You mentioned a couple strategies for increasing the concentration of the drug within the blood-brain barrier. Is that typically done in phase 2 setting or before or after? And what are some strategies that you kind of wait before doing that?
So it's usually done earlier because you want your efficacy study to be done at the best dose. Mm-hmm. So you play with what your dose is going to be in the phase 1 setting. But when you go into phase 2, it's not 3 or 6 patients per cohort, it may be 70, or if you're doing a randomized study, 150 patients. And it's really hard to play around with that. So again, there are all kinds of ways in which people have tried to do this before. Intra-arterial therapy. Intra-arterial therapy with hypertonic mannitol. But again, the regional treatment, the tumor does not confine itself with one blood distribution. So then you end up having to do the right side, the left side, and the back. And there are all kinds of toxicities associated with that. People are now looking at whether there's ultrasound that you can use to transiently disrupt the blood-brain barrier. We think the better technique is going to be these vasoactive peptides. Heard a little bit about it first, putting drugs directly into the brain with catheters. I mean, obviously, psychiatrists are interested in this, neurologists are interested in this, infectious disease folks, Parkinson's, docs. So there's been a lot of effort in the past to try to make this work, and it hasn't worked. So I think, I mean, if you just look at statistics, chances that our approach is going to work small, but if we're going to make any progress in brain tumors, you're going to have a good blood barrier.
And when you do the PK studies, has it ever been a higher concentration of the blood, or is it generally equal or less?
You know, there are some compounds that are cleared from the blood relatively rapidly, but not cleared from the brain so rapidly. So there's a possibility that we could do that. There's been a lot of work done on programs. They get pulled in by something, and it's out there. You know, so if you're looking for a good project. Yes?
You mentioned in the beginning, you know, how important the blood-brain barrier is, and I'm just wondering if you open it, what are the side effects?
Such a great question. If we were able to open it, we have to realize that we're not only opening it to our drug, the patient also has to be on lithium. All the lithium is going to go in there, and that's not going to be a good story. And there's a lot of drugs that we could not give if it was not for the blood-brain barrier. For example, vincristine is a neurotoxin. It's a chemotherapy drug that's used, but if it's accidentally injected in the spinal fluid, you die right away. There's no way to stop it. So if you ever got that drug to the brain, it would be a big problem. There are a series of papers that are really fun to read on the evolution of the blood-brain barrier. Insects. They have a distributed nervous system. But way back when, in evolution, they decided that in fact keeping nerves isolated from everything else that changes in the body was important. So their blood-brain barrier is neuromally based rather than endothelially based. as the brain gradually, or as the nervous system gradually sort of focused in one area. So the special blood vessels that came in, and the endothelial lining took over the normal lining's job. If you take a look at the evolution of fish, there are about 6 times where there was a conversion, separate pathways. There was a conversion from the neuronal barrier to an endothelial barrier. Sharks still have a neuronal barrier. I think that sends the message that speaks to the question you were asking. Hundreds of millions of years. Evolution has said you need to have an intact blood-brain barrier. If we screw that up, it's probably not going to work very well. So the approach that we're taking to open the blood-brain barrier is something that will be very transient. It'll last forever. And then it goes back up into its normal self again. I think if we were to try to open the blood-brain barrier and leave it open, again, evolution would tell us that that is not how it works.
You mentioned the drug thalidomide to treat the glioma, and I'm thinking that you mentioned the drug to treat the glioma, and how, when in the scenario, when you show the different curves and the comparison of them, how do you decide when to stop this treatment?
When to stop the temozolomide? So it's a great question, and as an oncologist, somehow more is always better. So we do studies saying you're going to give this much, and then there's a small effect, and we never go back and figure out which parts of that are really effective. So the study I showed you with temozolomide was published in 2005. It was designed 5 years before that, so 2000. We're now how many years from then? And we still don't know what the active components are. Some people are arguing that it's mainly the concurrent radiation of temozolomide that works, and that the extra 6 months of chemotherapy that we give these people is useless. I tend to agree with them. I think that's probably true. Other people have said, well, we need to give more and more chemotherapy. And there's some people who argue that we need to continue this not just for 6 months, but a year or 2 years. So the data we have so far is that increasing the intensity of the adjuvant treatment doesn't help. Increasing the duration of the adjuvant treatment doesn't help. So the next step is actually going to be to cut back and see whether less would be the same amount. And that might also reduce the duration of the lymphopenia that these folks have to back off on the chemotherapy. But the answer is we don't know how long we need to continue. People have argued that it is not worth spending research time and patience on answering those kinds of questions. We need better drugs.
I was wondering if you figure out how to open up the blood-brain barrier, then could you make the radiation obsolete so you don't need radiation and we only use chemotherapy plus immunotherapy?
So the best treatment for our patients right now is radiation. Chemotherapy adds to the good thing, but the radiation is the important part. So we have to make some big changes in our chemotherapy before we could suggest that. We have some other ways we can probably make the immune system healthier with radiation. I mean, the first step is to realize that we have a problem. Second step is to understand sort of why we have the problem. But I think we're getting there, and so now I think there are all kinds of ways that we have to think about reducing blood exposure. We built a computer simulator to map out with each radiation field what's the total body blood exposure. The radiation oncologists have never considered blood an organ at risk for toxicity, because it tends to flow by and they calculate all their stuff based on a single location. So I think in another 2 or 3 years we'll have that part of it all figured out. But of course.
Is radiation absolutely necessary? I mean, even if you can open up the blood-brain barrier, is it absolutely
So-
hold the baby for 1 hour and then use the chemotherapy and then.
So let me answer your question a different way. In lymphomas in the brain, the prior treatment was radiation, but there was too much side effects of the radiation. We now use high dose chemotherapy, and we don't give radiation. But that's a very sensitive tumor, and this is a very resistant tumor.
Thank you very much. That was very interesting and different than at least what the kind of clinical trials that I am involved in. Again, let's thank Dr. Grossman. And if anyone has other questions?
Yeah, I mean, so. PhD. Good.